PBL project on karyotyping.
To diagnose genetic disorders in humans, scientists use a technique called karyotyping. In this PBL, we simulated this procedure by using a matching model named Disorder Detectives, examining human chromosome disorders. To accurately diagnose our patient, we match chromosome stickers to the corresponding chromosomes on the booklet. We found an abnormality relating to his X and Y chromosomes, which is why he's having trouble bearing children. After this, we set out to determine how many men included in the whole simulation exhibited genetic abnormalities.
The contents of our PBL project include our background research, our methodology, our ELA and social studies component, and the conclusion.
Our background research consists of what karyotyping is. Karyotyping is a laboratory technique that involves the visualization and analysis of an individual's chromosomes. It's often used to identify chromosomal abnormalities and genetic disorders. You inherit your chromosomes from your parents. Normally, all of your cells, except for the egg and sperm cells, have a set of the same 46 chromosomes, the chromosomes being grouped into 23 pairs each, having 2 chromosomes, one coming from each parent.
Karyotyping is commonly used in prenatal screening to identify chromosomal abnormalities in the fetus, such as Down syndrome, Edwards syndrome, and Patau syndrome. Karyotyping is often performed on cells obtained through procedures like amniocentesis or CVS, which involve collecting samples of amniotic fluid or placental tissue. Respectively, karyotyping can be employed to investigate recurrent miscarriages. Chromosomal abnormalities in one or both parents can be assessed to determine if there is a genetic basis for the repeated pregnancy loss and diagnosis of fertility issues.
So here's our background. Our research question was, how does karyotyping work and how does it benefit the health field? Our problem was, how can scientists accurately diagnose genetic disorders? And we hypothesized that we would be able to accurately diagnose a genetic disorder using a karyotyping simulation. And the purpose of this project was to better understand karyotyping and how it works to diagnose genetic disorders.
Our methodology and our variables were our control variable being our cell sample collected. Our independent variable was matching the chromosome decals, and the dependent variable was whether or not the samples contain mutations.
Our procedure was to gain background knowledge on karyotyping, then pick one simulated patient to study, grab one chromoscan board to get ready and assemble the decals, and then randomize the chromosome decals to reduce predictability.
Then we stuck the chromosome decals onto the corresponding chromosome on the chromoscan board to note, and we noted any abnormalities. Then we made a diagnosis and checked in the booklet to ensure the diagnosis was correct. And we were done. We placed the chromosome decals back onto the panel in random orders for the next group's usage. And in this picture, you can see that our cases, X chromosomes, X and Y chromosomes, were mutated because we found 2 X chromosomes.
Our results. Our data showed that 5 out of 7 men that we studied showcase a genetic deformity that affects their standard of living. The case that we decided to study was study H, which further than others and found that he was suffering from Klinefelter syndrome, a genetic disorder that is only present in males and affects their fertility. For our conclusion, our hypothesis had been proven correct because we were able to accurately diagnose our patient using a karyotyping simulation and found the genetic deformity present in his sample.
Klinefelter syndrome is a slightly rare condition that is only seen in boys. Klinefelter syndrome affects the fertility because it has an impact on the size of the testicles and how much sperm they produce. People with this disorder produce little to no sperm, which explains why case study H is having a difficult time bearing offspring. His prior learning disability diagnosis is also explained because Klinefelter syndrome can cause intellectual delays in young boys. Researching an application of this disorder closely gives insight into how it affects people in real life.
In our social study component, we discussed where the name karyotype comes from, how it was discovered, who discovered it, the first time it was experimented, and the importance of karyotyping. Karyotyping helps expose chromosomal abnormalities and the impact it has on human health. It also goes into detail on how karyotyping came to be and the progress that it has made over the years.
In our ELA component, we explained how long it takes to get karyotyping results. It usually takes 1 to 2 weeks. What a karyotype test looks for, it looks for any abnormalities or extra chromosomes or missing chromosomes in your chromosomal chart. In order to get a karyotype test, you would need to donate blood, bone marrow, amniotic fluid, or placenta tissue, and your results would differ based on if you had any abnormalities or if you didn't, and we are looking for any genetic issues. In our experiment, we used a simulation, so we used a test board and decals. We assembled our decals, and after that, we put our board away. And our patient, we discovered, had Klinefelter syndrome.
Once again, we are students from Harmony School of Advancement. Thank you for listening to our research.